Okay. Thank you very much for the nice introduction. Thank you all for being here. So today we're going to talk about the biological signature of brain disease. I will try to convince you how this differ from the so-called biomarkers and other concepts about disease. And I will introduce you the Medical Informatics Platform, which is one of the tools developed by Human Brain Project to exactly provide this link between fundamental neuroscience, clinical research, and patient care. So the framework what we're working with is really in vivo study of patients. So that means we have our disposal very few little tools that allow us to go inside the brain and study what's happening when the brain disease occur. So the main tool that we are using is neuroimaging tools, and we have this proposal that in fact we can use neuroimaging really the same way that in personalized medicine we use genetic as a fingertip of the what's happening inside the brain. And by using mathematical modeling, we can try to see what are the link between changes in the brain and what's happening behavior. So my lab is involved in this work in the Human Brain Project. We are located within the hospital at CHUV, and we work closely with clinicians. And we also study normal population healthy aging. So the framework where we're located within the Human Brain Project is really how medicine can benefit from the development in the Human Brain Project in terms of neuroscience, how the knowledge that will be derived from the neuroscience understanding at all levels of representation of the brain will help in terms of medicine, and how we can use computing power to develop technique to understand brain diseases. So throughout the whole Human Brain Project is the idea that we need to bridge the different scale of representation of the brain. So when we're talking about brain diseases, as I said, there's very few data that we can collect about in vivo. So we can collect information about behavior, how a person is behaving, his cognitive profile or decline. Now, thanks to you can do neuropsychological testing. There's 100 years of neuropsychological testing that give you fine-grained understanding about what's happening in terms of memory, for example. And thanks to neuroimaging, you can go now inside the brain and see what's happening and map dysfunction in the human brain. So you can also collect information about genetics, about what's happening in the protein. But what's really important is that we try to link this different scale and have this biological signature of disease at the right level where we can develop treatment and diagnostic. And this is what I'm going to explain to you. So the Medical Informatics Platform is one of the 6 platforms of the Human Brain Projects, and is also tightly linked to the neuroinformatic brain simulation and other platforms. So our goal is really to first to say we need to collect data. I mean, in order to develop model and to develop this model derived from data, we need to collect data. And unfortunately, we heard a lot about big data analysis and this kind of thing, but there's very few data available as well in terms of medicine that you can use. Study, for example, in vivo study, you can consider that you have a large cohort when you have something like 50, 100 patients, because like massive cohort, right? Today, there's this large-scale initiative that tries to collect more and more data. We're talking about 1,000 maybe people, but it's relatively small, right? Still. So our idea was to try to collect data in a place where it is collected routinely, and this is in the hospital. So now the difference between the data that's collected in the hospital and data that you collect in research is diminishing. You have the same devices in terms of MRI scan. You have the 3 Tesla scanner that you find in the neuroimaging lab, state of the art. You find them in the clinics, and the protocol are more and more harmonized between the two. So there's a source of data there which is very rich that we can make use of. And also the data that you will find in hospital, they are also representative, maybe better representative of the disease and the population. Just to give you an example again, when you do a study, for example, research study, you want to study, let's say, Alzheimer's disease. You will have your exclusion and inclusion criterion. And for example, you will select people that have Alzheimer's disease but nothing else, right? When you go to the hospital, you'll find that patients that have Alzheimer's disease, that have a history of stroke, they have history of depression, they have comorbidity, they have all this sort of thing. That might explain the difficulty to translate research to clinic because of these differences in population. So we need both high quality research data, but also study the population which is more representative of what's happening in the disease. There is certain difficulty in trying to collect this data in different hospitals in terms of privacy, but I will talk today about the use of machine learning technique and to develop this biological signature of disease. What we try to build as well, because it have to be like an effort between different communities, and what we try to build is give access to researchers to this data that will develop data analysis, hypothesis testing, and the same way clinicians can use the same tool for decision support. And everybody who's able and interested in this type of data can also contribute, and also targeting the general population is something that is very interesting because the brain disease affects a lot of people, and we often have people that are interested in contributing, and we allow this in the platform. So the critical thing is how to go beyond symptoms. So today the way to diagnose disease is based on symptoms. So Alzheimer's disease, an effect of memory, and you have change in behavior, change in daily life, right? That's the clinical definition. In many diseases, you have this sort of clinical decision classification, which is based on symptoms. And this is not really useful because symptoms are not discriminative enough. Having a problem of memory is not discriminative enough to give you a really accurate description of the disease. So we want to build something like which is very important in medicine, which is evidence-based medicine. And an evidence-based allow you, by collecting a lot of data, to do very interesting things. It's not just increase in power and size, is that you have, because you have more sample size, you have a better estimation of the effect size. This is very important. You can also study consistency. That means if I observe a certain phenomena with within certain modality, is it replicated in different modality, for example? And this consistency gives you an accumulated evidence. You can try to touch on things like causality, because you have temporal information, longitudinal information with this hospital data. You can start to think about biological gradients. If you have multiscale information, you can think about replicability. You can develop technique for clinical trial and think about alternative hypotheses. So this is the way where we can move from symptom to biology and move from just an observation to causality and experiments. So now, what is this concept of biological signature of disease? So as I said, we can collect information about behavior, neuropsychology, neuroimaging. You can collect information about genetic and protein. And unfortunately for us, the information that you want is what's happening at the cellular level, molecular level, because it's where you want to target treatment. And this will never be accessible, or up to now is not accessible in vivo in human. So we have to sort of reverse engineer and try to guess what's happening at this level just on the basis of the information that you have outside this level. So the observation, what we call the phenomenological space or the disease space, is the observation made by clinicians. Is about behavior, about memory, about learning, so on and so forth. At our disposal, we have the multimodal observation, brain imaging, genetic, protein measurements. And up to now, people were working sort of silo. You have study of disease, which are, let's say, like in our lab, for example, we are biased toward brain imaging. So this gives you a certain picture, but not the total picture. And you have people who start with a certain gene and try to find the link between often one single gene to disease. So the first principle of this biological signature of disease is that we need to take into account these different modalities first. And we believe that there's sort of a causal feature, which is this intermediate space, which is the combination of these different phenomena happening that you measure, your observation, brain data, genetic data, and protein. And the first thing we try is to develop methods that explain this data in terms of variability and variance. So there's a lot of statistical methods that you can develop, try to explain just the data, and in terms of summarizing this data. And the second constraint is that you want this feature to be predictor of what's happening at the behavior level. So this is the model, sort of mechanistic model that's linked observation to different scale of observation. And I will give you a few examples later on. So we had to build this platform. I will give you a few hints about the architecture, and we'll see later on some example. So the main goal was to collect this data from different hospitals and combine it from a research cohort. And this platform allows you to do disease signature by using data mining in real time, visualization. You can ask different query questions, and more focused hypothesis testing and model-led approach. So we have what's called a federated approach, where we have different, the data are in different places and are collected in the central place. So this is the web base where you can ask these different questions. That poses certain challenges in terms of what kind of thing you can do if you're not allowed to move this data from different places and they're distributed. So this is sort of another level of complexity. This is sort of busy slide, but just to explain you. So we're here at one place, let's say one hospital. We can collect data from MRI brain, clinical information from the electronic health record, and genetic information in some of the our partner hospitals. So we have a server which is called the Data Factory, which in fact already bring state-of-the-art research tool to the hospital. That means we're going to, for example, from the MRI scan, extract brain features, gray matter volume, cortical thickness, connectivity measure, all this state-of-the-art technique. So already from this, we are making this qualitative information made by radiologists to quantitative measurements. This information is put in a database, and we have a second server which implements all these machine learning tools, which we call the algorithm factory. Is going to run either in real time or triggered by the user these machine learning tools. And data will be federated at the central level where user can access the platform. So why this is important, not just to collect data from hospital, but research data. We have sort of framework where we can use the high quality research data to develop, like I say, a biological model. For example, I can develop a generative model that's linked the brain, what's happened to the brain, to Alzheimer's disease, for example. And there's many of such models developed with high quality research data. But the question now, I can test this question about the validity of the model in the clinic. With this infrastructure, this model can be tested with the hospital data within each hospital. So we use this sort of virtuous circle between model validation, causation, and improving our model directly on the relevant data. And you can also improve the quality of data by using high quality research data. So the type of methods that we implement, as I say, is quantitative measurement, where you extract measure from the brain anatomy using computational anatomy tools, and we'll see in the tutorial how we can do this. And then you extract measure of volume, thickness, etc. And you can apply univariate methods, which will look at single voxel in the brain or regions at the link between the clinical score, behavior, and other thing. You can do classical statistical inference, or you can apply more sophisticated methods, statistical learning methods, if you have the idea to separate certain disease categories. The data. So, as I said, one of our first goal is really getting access to data. This is very important. So the data, what we call the data factory, is about transforming the data that are available somewhere in a disk, in the storage, in a hospital or research center, to something valuable for research. And we have all these different processes that will transform this data in useful measurements. And we are developing sort of a description of this data in terms of variable ontology that's really linked to already to the biology. Is it genetic? Is it molecular? Is it related to the system? And if it's related to cognitive system, which system? Is it motor? Is it memory, etc.? So that's an example. For example, when the variable arrive here, they get categorized in different domain and different scale of representation. So this is for the first part of this of my talk. So the Medical Informatics Platform was launched in March. Is available. You can log in and already do some analysis. There's already some research data and hospital data in this platform. So this is the end of the first part. I will show you some application. But do you have any questions to know? Yeah? Just before it. Yeah.
Regarding patient recruitment, what is sort of the, is it an opt-in process or an opt-out process, or is it changes from country to country?
So the depends on the places in the country. So in majority people had university or hospital, they have this sort of opt-out in general. But now, you know, it's better to give people information. People want to be informed, so it's more like an opt-in or very informed, let's say, opt-out. And then the way to improve recruitment is always to talk to the people. And when you talk, and it's the reason why people are often like to volunteer. And in our experience, in our hospital, for example, we went from 50% people agreeing to 80%. When you really explain your research, people do want to volunteer and give their data. Okay. So I have here a little video about the, I don't know. I don't know if it's going to work. Okay, I will play this way because it doesn't work full screen, and I will be talking over it. So this is the Medical Informatics Platform. So one of the, let's say, key concept that we have, because it was actually a constraint, is that we cannot give the data to researchers. So everything is within the platform. You query the data in the platform, you do the analysis in the platform, you get the results in the platform. So we were at the point where we say, okay, why not write in the paper in the platform? And so basically you can write paper in the platform, and the idea is to foster collaboration. So you do one analysis, you build the model, and then you write it up. Yeah, there's always a link that will link this paper to the data used, the method that you use, and to your own results. And somebody else can come along and change this model, modify it, and incrementally we improve the understanding of brain disease. It's very sort of collaborative approach. And this is what you're saying here in this video is what I saw about the data model, the representation, the extraction of different variables about brain structure, about behavior, etc. They appear in sort of, instead of pull-down menu, this sort of bubble nested structure. So you can already ask a question about differences between different type of disease. For the moment, it just concentrates on dementia, but we'll be adding on different type of disease. And then once you have identified your variable of interest, for example, I want to see the link between what's happening in the hippocampus and for example, memory, I can choose these different variables, and I can build the model that will include this variable. I can include the interaction with the genetic factors, and so I can evaluate this model. The methods for evaluating this model can go from classical inference technique, sample t-test, linear regression, ANOVAs, to more sophisticated, let's say, machine learning techniques where you want to make predictions. Yeah. So this is the video showing you how you can interact with the data and save your results. So I will skip quickly here. This is the machine learning part where you see, the model is saved and made available for other users. So let me go back to my this video is available online on YouTube. You can see the different demonstration, but I will give you one example, which is pertinent. So in this example, we try to replicate a study, and this study is very interesting because you can look at it from 2 different points of view. You can look at it from the research point of view. I'm interested in the mechanism of Alzheimer's disease, and one of the known hypotheses is the beta amyloid hypothesis. So from research point of view, I'm interested in this, and my question is about the effect of one of the main gene risk factor, which is ApoE4. So this is from a point of view of research, it's in interactions that I want to look at, and I can use the platform to study this question. The same question, if I was a clinician, for example, the way to measure this beta amyloid, for example, is to do a PET scan. And the PET scan is something what in some places you get reimbursed, in some places not. Because for different reasons, some places think that is not helpful for diagnostic yet, and other places, and it's very expensive. So I can collect information. What are the evidence that this PET scan is informative, and I can, for example, use this as an argument to get the patient reimbursed. So the same question, different perspective. And then this is a paper that was published that you can replicate because you have the same data in the platform. So in the platform, you will query information about, okay, my question of interest is about Alzheimer's disease. I will choose this as a diagnostic. I will choose a variable PET in some regions, whole brain or different subparts, and I will select ApoE4 as a covariate, and I will do the interaction. And you will have such plot that show maybe convincingly or non-convincingly, depends on your belief in statistics, that there is differences between patients that have the risk gene or not, and these differences is shown on the PET scan. So that will be the type of question you can ask. Other type of question, there may be more like a, let's say, black box question. You're not that interested maybe as a clinician, maybe about what's happening, the mechanism, but you want like a diagnostic tool or something that summarizes the information. So you have the tools that are like a classification tool, do binary or continuous classification tool like supervector machines. And this is an illustration from a paper published by our group, for example, where we compare the difference between machine learning tool, supervector machine that only have access to one image versus the best clinicians in the world at the Queen Square. So you can read the paper and you see who is better. The machine is not doing bad. Actually there's only one clinician doing better than the machine. But the clinician had access to the whole history of the patient. So on the basis of one image, you can have quite accurate diagnostic. But again, I would say there's 2 caveats to this. First, if you remember what I said in the first part, the diagnostic is mostly, because it's based on symptom, is mostly not correct. So if, for example, in the case of Alzheimer's disease, which is well-known disease and well-characterized, the error rate is 30%. So that means if we do post-mortem analysis of this brain, we discover that in 30% of the case that people we thought they have Alzheimer's disease, they didn't have Alzheimer's disease. Either they have something else or something they have nothing wrong with their brain. So, and you work in both directions. You have people that live healthy all their lives, show no sign of anything, and in 20 to 30% of the case, post-mortem, you find traces of Alzheimer's disease. So this is why it's very difficult to study, because you have such an error rate of misclassification, 30%. So this type of tools is not really to replace doctor or to give like pure diagnostic, is more to give certain evidence again. We're talking about accumulation of evidence. Another example that we did in early stage was to answer the question, does it really make sense to combine all this data from different modality, right? You have PET measurement that are different scale, you have MRI from different scale, you have the clinical measurement that's a categorical, maybe wrong, you have genetic, and when you pull all this together, because the data is not in measuring every subject, you have also missing information. So this is something that's usually a statistician you not do, usually like putting everything together. But here we try to test this hypothesis as a proof of concept, to say we can put all this information together and achieve something in terms of understanding brain disease. And in this case, it was Alzheimer's patient, and we say, okay, let's assume that in fact we don't have one type of patient of Alzheimer's disease, but different subtype. And let's assume that even for the what we call healthy control, in fact, there's different subtype. There's people that might be that might have disease, but in fact they don't manifest it. You have people that are super healthy or super normal, etc. So let's assume now we do this sort of thing. So the approach will be to use some sort of unsupervised or semi-supervised clustering, because you have this information about difference between two groups, but that's it. And you want to find a different subgroup. So when you do this with this data, we identify more than 10 subtypes of Alzheimer's disease. And the biggest one was the classical that you find in textbook: atrophy in the hippocampus, problem of memory, textbook. But you find also different subtypes that doesn't fit in this descriptions. And more interestingly, we find also more than 10 subtypes of normals. And they were well characterized in terms of genetic profiling, in terms of protein, and they're really different from the rest. So this is a way to describe a population in terms of its variability, and this is a way where we go towards what's called personalized medicine or precision medicine. So personalized medicine is not about one individual, it's about finding these subtypes of different individuals that are homogeneous in terms of their biological description or profile or clinical scores. So the next step will be to say, okay, I have now this homogeneous subgroup. Does it behave the same way longitudinally? What are these people that we identify this normal with already some atrophy? Are they going to convert to Alzheimer's disease at certain time points? The third example that I will show you is how we can combine informations from different scale. So in this example, we are lucky enough to have a very small data set, very small, just 33 subjects, but very rare and very useful. So these subjects, there were people for which we had post-mortem validation. That there were people that have cognitive decline during their life, and post-mortem they had sign of Alzheimer's disease. So post-mortem quantification. And we're lucky to have the MRI scan before the death. So with this, we say, okay, now we have maybe let's say a gold standard, a better data set to train the machine learning methods. So we use this to train a machine learning approach based on super vector machine, and we use the pattern that the machine learned to classify and two other independent cohorts. Right? And in these two cohorts, the patients were classified based on clinical diagnostic. So you have patients that were classified as Alzheimer's, some other classified as healthy, and what we identify is that although there was an overlap between the clinical diagnosis and the one made by the machine, we identify subtype of, of healthy control that have no symptom, because this is why they will have defined as healthy, but they have pathology. They already have atrophy in the brain. And then when we do follow up, we find that these people that are higher risk to convert to Alzheimer's disease. So this is the important thing to identify as early as possible these people before they develop the symptoms, and this is important for clinical trial. I have many examples. Okay. A fourth example is the use of deep learning algorithm, and here the question was very focused. It was not to apply just deep learning algorithm for applying deep learning algorithm. It was to say, okay, was to help us to try to identify which are the best features. So now we're collecting more and more data. We have a lot of features, and we want to use this method to select features. And we compare two type of deep learning algorithm with two different sets of features. In one side, we try to be, let's say, very smart, very clever, and to say we're going to inform the features. So we use region-based segmentation based on our knowledge of the brain, and we take different region, and this was used as an input for the algorithm. And in the other one, say we're maybe less smart, let's just take random patches throughout the brain. And what was interesting about this is that the results were exactly the same. So the ability of the machine to identify which are the best patches was the same as that if you put your best knowledge in terms of which area will explain better the disease. So we were quite happy with this result in terms of how we can use this method for better selecting features. So this is almost the end of this presentation, and I present to you, in one hand, the concept of this biological signature of disease, the combination of different scale information, and our aim to try to reach to this level where we can have description of disease more and more linked to biology and not to symptoms. On the other hand, I show you the more data-driven approach, machine learning techniques to derive diagnostic. But between the two, I think it's important to develop models. This is very important to have disease model that's linked really to different scale. What you want is a parameterization of your observation, that you're able, for example, to replicate it, you're able to tune, and this is more useful as a future framework. And the main idea is that the main challenges, I would say, this is how to combine, how to go from one scale to the other. And again, I think neuroimaging can play a role because we have this observation, for example, from fMRI or from PETs, what I showed before, from EEG, DTI, that allow you to go from different scale. And now we have a framework that link fMRI to EEG, DTI, and so on. We need to go beyond and link this to what's happening at the neural level in terms of cellular level. And this is one of the biggest challenges. The next challenge is the link between neuroimaging and genes, gene in general, or gene expression for personalized medicine. So in the context of brain diseases, neuroimaging is very personalized. We're already making decision about which treatments, which course to take based on the neuroimaging. So this is the same way that's happening for personalized medicine in terms of gene. And the combination, that was one of the what we learned from our study by combining all the information, is that in order to do GWAS or gene expression analysis, you need to have a lot of data. And what we find out very interestingly is that the interaction between neuroimaging and gene give more power to the analysis, because neuroimaging is quite powerful. You just need, for example, if you compare 10 patients versus 10 control, you have very strong signal. So the combination between the two is very interesting, and then we want to combine these two in very sort of smart way, where they can borrow evidence from one and the other, and the way to do this is to develop Bayesian modeling. So Bayesian modeling is the way that in fact you can introduce you this domain knowledge. Then in the example that I show you where we combine all this data, it was a bit, let's say, simplistic, because all the information they are flat. You just put the genetic information, the brain imaging, the protein. But what you want to put is more to know how each information is has an impact on the other, and you have more like a hierarchical model. And this is more interesting in terms of developing this disease modeling. So I think this is the end of my talk. I will show you the clustering to finish here, and thank you for your attention.